Hello, welcome back. This is lesson 4, and in this lesson we're going to talk about regulation in glycolysis. Now in lessons 1 and 2 we already discussed glycolysis, so you should know what glycolysis is by now and the reactions in it. If you didn't, go back and try to go over it. However, if you already understand glycolysis, let's get into the regulation. So what is regulation? So regulation is when you control the activity of an enzyme by external factors. So any factor or any environmental signal that can change the activity of an enzyme is regulation. So you're basically regulating the enzyme based on how it's supposed to behave. And regulation is very common in cells. You need regulation to survive. If a cell is not regulated, if something is overactivated, underactivated, there could be many issues. For example, in cancer cells you have overactivation of certain enzymes, which leads to uncontrollable cell division. Obviously, it's more complicated than that, but basically, no regulation means bad. So, if no regulation, it's bad. So, in glycolysis, obviously, we have regulation. And some ways we can achieve regulation are the following. So, firstly, we have allosteric regulation. Allosteric is when a molecule binds to an allosteric site. So you have an allosteric site, and a molecule binds it, causing a conformational change in the enzyme, and now the enzyme has a lesser affinity to what it is supposed to catalyze. Another form of regulation, which is also pretty common, is covalent regulation, and this means having covalent binding of molecules, such as you can have covalent binding of phosphate. So you have phosphate groups. You can have methyl groups. Another way is hormonal. When hormones act on receptors and through a signaling cascade, you eventually get some reaction. And then this, let's say, activates some enzymes, and these enzymes can, let's say, do covalent modifications such as kinases. So obviously these regulatory pathways have to be connected to one another. Some other way could be acidity. So some enzymes do not function well in certain conditions, and acidity can actually regulate the conditions. And we're going to talk about a specific condition in glycolysis. Now, another way can be transcriptional regulation. So you have the expression levels at the transcriptional level, right? So on the DNA, you can have sort of regulatory signals, such as that the more transcription you have of the segment of DNA, the more of the transcript you have, and the more enzymes you produce. So if you want to downregulate activity, what you can do is you block transcription and you reduce the amount of enzymes, and therefore you reduce activity. And this can be a form of regulation. The regulation functions differently in the muscle versus the liver in terms of glycolysis. Obviously there are similarities, but in this lesson we're going to talk about in the muscle, and in the next one, part 2 of regulation, we're going to talk about regulation in the liver. Alright, first things first, the steps I'm showing here are the irreversible steps. So if you remember in glycolysis, this was step 1, this is step 3, and this is step 10. So I've skipped here, so this goes from steps 4 to 9. Those are not important, those are reversible reactions. However, these steps, they're irreversible reactions, so they matter. I included this one because this actually has to do with regulation, and this is a reversible reaction, so let me make this clear. And let's start off with the muscle. Unlike the liver, is not involved in blood glucose regulation. So in the muscle, regulation mainly depends on ATP and AMP ratio. The more ATP you have, it means the more energy you have. Therefore, it inhibits certain enzymes. So in the muscle, it depends on the ATP versus AMP ratio. Let's say you have a high level of ATP or a high level of the ratio. So if the ratio is high, right, it means you have more ATP and less AMP. So therefore the ratio is high. So what this does, because you have a lot of energy, is a signal for having high energy. Therefore you want to inhibit glycolysis and produce less energy. Therefore ATP binds to PFK-1 allosterically. So this is an allosteric regulation. So it binds to it. So ATP goes and binds to an allosteric site. And now PFK-1 has lower affinity to fructose 6-phosphate, and therefore it does not produce fructose 1,6-bisphosphate as much. Therefore the enzyme activity is reduced. So this is inhibitory. Here on the left, this enzyme is actually PFK-1. So it's phosphofructokinase. However, it's just one subunit. And phosphofructo-1 has 4 subunits. And what it looks like as a whole. But let's focus on one subunit so you get a clearer picture. So in this picture, you might recognize this molecule from the previous lessons. This is fructose 6-phosphate. So fructose 6-phosphate. So this one here is the 6th carbon. Therefore you have a phosphate group on it. This is the first carbon behind here. This is the second carbon, third, fourth, fifth, sixth. And these two molecules are ATP. They are ATP molecules. So this is ATP, and this is also ATP. So you can kind of recognize that there are two sites here. So these have bound to the enzyme at two different sites. So in this site, you have the ATP and the fructose 6-phosphate. And remember, in the third step, and even the first step, we used ATP. We used ATP, and we formed ADP, and we added the phosphate group. So in this step, you needed ATP. And that is why you see an ATP bound, because this is the active site. So this is the active site. It's the active site. At the bottom here, it's just a binding site for ATP. And as we discussed, ATP is inhibitory and it binds allosterically. So therefore, this site is an allosteric site. Or in this case, we can call it a regulatory site. Because, as I said, the enzyme consists of 4 subunits. And the subunits, there's the M, there's L, there's also P for a platelet, so muscle, liver, platelet, and obviously different combinations can be found in different tissues. I want to show you real quick. So if we go down here, this is the full enzyme, which consists of 4 subunits. So imagine in one subunit, there's 1 regulatory and 1 active site. So in one enzyme, you have 4 active sites and 4 regulatory sites. And to give you a clearer picture, what I just showed you is basically around this much. So the active site would be around here. The allosteric site would be around here. So if you count all of it, you have like 4 here, and you'd have some active sites here. And obviously I'm not accurate with this. It could be approximately where I drew them, but I hope this gives you a clear picture. And a lot of enzymes function in a similar manner. Now let's go back to our discussion. We discussed in the muscle, high ATP ratio, AMP ratio, it's gonna inhibit PFK-1. Another thing that inhibits PFK-1 is acidity. So if you have a high H plus content, so let's say H plus, and you have a high amount of it, so increased because of lactate or lactic acid, right? It's a byproduct of anaerobic respiration. It's actually a product. I wouldn't call it a byproduct. However, increased acidity in the muscles basically inhibits this enzyme. So that's another inhibitory signal in the muscle. So increased H plus. Also, you can say a decrease in pH. And also, the opposite way works. So a higher AMP ratio. So if the ratio was higher for AMP, So if the ratio was lower, right? If you have higher AMP, lower ATP, the ratio would be lower because the denominator is higher. And this, on the contrary, would cause a positive effect. So it would actually bind allosterically as well and cause the activation of the enzyme. So it would change the conformation in a way that the affinity of the enzyme to fructose 6-phosphate is increased, and therefore you have more of the reaction happening. And why AMP and why not ADP? And this is because ADP still has some energy. So ADP has higher energy than AMP. Therefore, AMP as a signal is more sensitive, because at that point you know for sure the muscle does not have energy, right? So you activate it. Whereas ADP still has energy, and of course ADP has less energy than ATP. So it would still be a better signal than ATP to say that you don't have energy. However, AMP gives the best signal. So AMP gives the best signal. Moving on, the first reaction catalyzed by hexokinase is also regulated. So in the muscle, glucose 6-phosphate, the product of the reaction, actually inhibits it. So it inhibits its own reaction. So it's a negative feedback mechanism. Inhibits hexokinase allosterically as well by binding onto an allosteric side, changing its conformation, and now the enzyme has a lesser activity. However, you cannot regulate hexokinase with ATP ratio, ATP-AMP ratio, because glucose 6-phosphate is an intermediate for other reactions. So it is what? It is an intermediate for other reactions. Unlike this step, because this step, the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, is the first irreversible reaction unique to glycolysis. So this reaction, right, to this whole thing, is actually unique to glycolysis. Therefore, a lot of regulatory mechanisms from glycolysis work for this step. They regulate this step. And you're going to see later in the next lesson, in the liver, there's a lot more regulation going on, especially at this step. And lastly, we have regulation of pyruvate kinase. And pyruvate kinase, similar to PFK-1, is regulated by the ratio. And a higher ATP to AMP ratio means you have inhibition. So it's inhibited. And you could also say a lower ATP to AMP ratio is not inhibited. However, not because AMP binds to it, but because ATP wouldn't bind to it, so it wouldn't be inhibited. And lastly, fructose 1,6-bisphosphate actually plays a role in regulation by activating pyruvate kinase. So fructose 1,6-bisphosphate activates pyruvate kinase allosterically. And this is called, and this has a name, and this is called a feed-forward stimulation or communication. And what this means is that a molecule upstream from a pathway sends a signal downstream that, hey, there's a bunch of me up here, we're coming down, and we want you to be active because you're going to deal with us soon. These are all the allosteric regulation. There's also a communication between these two enzymes. And I want to go over it very simply. Imagine this. You have a high amount of ATP-AMP ratio. And what this does, it inhibits PFK-1. So when you inhibit this, what happens? Well, fructose 6-phosphate concentration increases, right? Because you're not converting it as much. And when this increases, equilibrium favors glucose 6-phosphate conversion, right? So you increase glucose 6-phosphate. And what this does is it inhibits hexokinase. So inhibition of PFK-1 actually communicates with the first reaction to inhibit hexokinase. And with all this regulation going on, the cell, especially in the muscle, so muscle cells, what they do is they function according to needs in the cell, and those needs are energy needs. whereas in the liver, it's based on how much glucose is in the blood.